Before we move on to this afternoon's seminar, I would like to provide you all with an update on our regular MC, Professor Ruth Grant. Sadly, Ruth has made the decision to step down as host of these seminars, effective immediately, due to health issues that she is undergoing treatment for. She's very sorry that she couldn't be here to say this herself and say goodbye to you herself, but she's asked me to let you all know that she's enjoyed meeting you and speaking with you over the years, and she would like to thank you for your continuing support in participating in these seminars, which she holds very close to her heart. On behalf of our advancement team here at UniSA, I would like to publicly acknowledge how grateful we are to Ruth for her commitment and contribution to these seminars over many years. She's hosted these events since 2013, so that's 11 years, and we warmly refer to her as an honorary member of our team here at UniSA. I'm sure that you'll join with me in wishing her the very best at this difficult time and sending positive thoughts and prayers her way. Now on to today's topic, which is about the development of new advanced therapies for healing chronic wounds. We are delighted to have two presenters here today to speak with us. Our first speaker is Professor Allison Cowin. Allison is a Professor of Regenerative Medicine and the Deputy Director of the Future Industries Institute at UniSA. She's recognised as a leading authority in the area of wound healing and scar formation, and she has over 190 peer-reviewed papers, editorials and book chapters in international journals and books. Allison is a Fellow of the Royal Society of Biology and Wounds Australia, co-chair of the National AHRA Wound Research Initiative and a member of Wounds Australia National Research Committee. She was the founding president of the Australasian Wound and Tissue Repair Society and former editor of Wound Practice and Research. We're very grateful to have her here today. Allison is joined by Dr. Xanthe Strudwick. Xanthe is a Senior Research Fellow, Regenerative Medicine at UniSA in the Future Industries Institute. Xanthe was awarded her PhD from UniSA in 2016 for her investigation of the role of the cytoskeletal protein flightless eye in tissue regeneration. That sentence had to be practiced several times. We got there. This protein is present in high amounts in both burns and diabetic patients and seems responsible for impaired healing. Xanthe is keen to shape future clinical practice and is working to improve the health of all Australians with chronic wounds. Please join me in welcoming both Allison and Xanthe today.
Thank you for the introduction. I'm going to slightly change the program. So we decided to do a tag team. So I'm going to set the scene, let you know why we're interested in what we're interested in, and then Allison can come in and tell you how we're solving it. So hopefully that all works for you. So as you saw in the first slide, we are going to be talking about that bench to bedside progression in how we're developing advanced therapies for healing chronic wounds. And we thought we'd get started by saying, well, wounds, what's the problem? We know the old adage, time heals all wounds. Actually, no, it doesn't. And even if it does, probably not very well. Is that loud enough for everybody or is, yep, good, thank you. No, it's not. Okay. Well, let's turn this one off and I'll just stand over here. Is that one better? Okay, so the lapel mic is not on. So the reason why we're interested in wound healing is because it's a global problem. So if we think of chronic wounds, these are wounds which fail to heal. So they're open for months. This is a massive problem. Worldwide, there are 20 million people who are suffering from a chronic wound. These can present as things like leg ulcers, pressure injuries, and diabetic foot ulcers. But added to this, we actually have acute wounds, those wounds which occur intentionally from surgery, or they might be from lacerations. They might occur because we're having cosmetic procedures or skin grafts, and there's 300 million people with an acute wound at any one time. We also add on traumatic wounds. Now, these don't just affect that sort of surface, our skin, but they actually, because of their size and their severity, they actually have compounding problems for your entire body. These traumatic wounds often arise from burns or other trauma, such as field injuries in war. So when you put it all together, it's a massive global problem, but actually it hits close to home. So within South Australia, we also have very high numbers. In Australia, there are 450,000 people suffering from a chronic wound today. In South Australia, that means that we've got one person every 5 minutes who's developing diabetes, with 8,000 people having been diagnosed in the last 12 months. When you think about the fact that we also have 180,000 South Australians who are actually at high risk of developing type 2 diabetes, you can see how in the years to come this is only going to expand further. Unfortunately, while any wound has the potential to become chronic, that is, it's not going to heal, the really significant proportion of these do come from patients that have diabetes, which is why we're so interested in this. 1 in 4 people who have diabetes will experience a chronic wound in their lifetime. And of these, 1 in 5 will actually not heal at all, such that they require an amputation because they can't salvage that limb. This is a huge financial cost to our healthcare, but actually personally the cost is far, far greater. Unfortunately, within the next 5 years, you have a less than 50% survival rate if you've had an amputation. So we're not looking to let these chronic wounds occur. We want to get in there earlier, and you'll hear some more about that from Allison later. So we mentioned that time heals all wounds. Well, for chronic wounds, it doesn't. They stay open. And if we think about what the role of the skin is, it is that first line of defence, that barrier to the outside world. So when you have an injury, you have a hole in the fence. If we had regeneration, which is what my focus is, what happens after you're healed is exactly the same in looks, but actually also in function. We don't usually get that. We get a hasty repair job where it's actually visibly and functionally different. But in something that's like a traumatic wound, like a burn, where there's really high inflammation at the start, it actually also ends up with a really bad scar at the end. The problem with scars is that they can actually be vastly different to what the original tissue is like, and it can also extend out of the original bounds of that wound. And if it's over something like a joint, you're going to have reduced mobility as well. And it definitely looks like the unbalanced immune system is what leads to excessive inflammation. So that's that hot redness, really uncomfortable feeling, and that is linked to scar formation at the end. So this is the 2 ends of the spectrum. You have wounds that fail to heal at all, or they can go too far, and they also have a problem. And we're interested in understanding why. I will also briefly introduce you to what is arguably the worst disease that you've never heard about, epidermolysis bullosa. This is a rare genetic condition which causes fragile blistering skin, and this is because there's a mutation in the protein, which is effectively like the glue that holds the layers of the skin together. Kids which are born with this disease get blisters from minor injuries, even from heat or rubbing or scratching, which means they are constantly in a cycle of wounding and trying to heal. In severe cases, these blisters can also occur inside their body, in the lining of their mouth and their gut. This disease is really interesting to us because it presents those hallmarks of impaired healing, which I was just talking about. Excessive inflammation, chronic wounds which never heal, and for those that do, they end up with fibrosis and scar formation. What this means is if you look at on the right-hand side, these are the hands and feet of a young child. But because of the sheer amount of friction and blistering that they've occurred, they have now formed into scar tissue mittens, so they lose that function of their hands and feet. So why does this all go wrong? Well, this is what we're studying. The normal process of healing we like to think of as a really clearly defined process where you have the first phase, which is called hemostasis and inflammation. Effectively, what that is that the first thing that we need to do in the body, if you've got an injury, is we need to stop bleeding and prevent infection. So that's that role. You get sort of constriction of your blood vessels, you get that clotting factor, and then a load of immune cells will come in to search and destroy. They're wanting to get rid of any potential pathogens, any bacteria which are there, and also clear out the dead and dying tissue. When this works well, that gets resolved fairly quickly and you move on to what's called the proliferative phase. Proliferation is really about providing all of the new cells to come in and replace that tissue. There's a range of different ways that this happens, but usually what we're going to do is we'll have an initial formation of the new dermis. You'll get reepithelialization, which is that closing of the fence. And then after this, underneath there, you'll continue to get remodelling. So that's where the potential for scar formation can occur because we're going to continually try and clean out and clear and make a better formation of the skin underneath. But to get it right actually requires a really tightly regulated balancing act. It doesn't always progress across. On the right-hand side, effectively what we're seeing here is that each of the different parts of that process all have different timing. And what we know is that when you have a longer, more severe inflammatory process, you have a higher risk of fibrosis. And that's mainly the focus of where we go. So why are wounds still an issue? Well, they're complex. And we like to think that they're multifactorial, right? There's so many different cell types that have to come in and work together to get this working effectively in the right timing. But it's impacted by factors that are both within your body and external. What underlying pathologies have you got? In diabetes, we know that there's also reduced blood flow and nerve sensation at the lower limbs, which is what is leading to those development of those chronic wounds on their limbs. So where do we go from here? Well, in terms of innovation, that's what we really need. If we look back over the last few hundred years, the first innovation was that we're just going to start cleaning things with cotton wool, and then we discovered iodine was quite effective at preventing infection. That actually was a critical change because we could actually stop a lot of deaths after minor wounds. Moving on, around the time of the wars, we got some advances in dressing, so we had the Band-Aids. You then see the rise of antibiotics, which can be given systemically, so throughout the body to treat infection, prevent sepsis and the dying from large infections. And then more recently, there's been a shift to more advanced wound technologies, which are about dressings which can be activated. And if we have a look now, we know that the advanced wound care market is actually worth about $14.5 billion worldwide. And this really is those over 3,000 wound products, which are all about how do we help heal a wound once it's occurred. They can be a whole range of different things, and we do work on this, which you'll also hear from Allison later. But also there are technologies. So you might have heard of vacuum therapy, which can help heal chronic wounds, or hyperbaric oxygen, which again is trying to provide some of those factors which might be missing when there's impaired healing to try and push things along. If we think about wound research, this has really increased in the last 30 years. So the number of papers in 1990 was about 16,500. In 2020, we're looking at 65,000 papers. And the reason why we're still researching this is because the more that we know that we don't know anything, and we need to try and find the best ways of healing these wounds. Wound research in 2024 looks a little different to what it used to. It's no longer led in the hospitals by surgeons. It's a team effort. We have cell biologists like myself working alongside of biochemists, physicists, and material scientists, or nanoengineers, engineers that work with really tiny particles, because we're trying to make smarter therapies if we apply them onto the top. It's industry focused and it's end user driven. And that is the focus of our regenerative medicine group out at the Future Industries Institute. Led by Professor Allison Cowin, who will speak next. There's a team of research scientists, including myself, and a number of PhD students who are also working on projects which are all feeding into this. So I will hand over to Allison now to have a chat about how we are addressing this.
Thank you, Xanthe, and everybody can hear me. So Xanthe has sort of painted the picture about how significant wound healing is, and the fact that there are thousands of dressings and products out on the market, but yet we're still not able to heal wounds. So what we're doing is to develop out at the Future Industries Institute, which is based out at Mawson Lakes. We're looking at various different ways of being able to heal wounds. So we're looking at therapeutics, new advanced dressings. We're looking at developing new diagnostics, so we can actually identify people who might be at risk of wounds. And then we're also developing new technologies. And I just want to give you a bit of an oversight on just very briefly and very quickly through these different approaches that we're taking. So the first one is our research into wound therapeutics. So over the last 20 years now, pretty much, and Xanthe was the start of that project. We've been working on this protein called flightless, and it was first identified in flies, which is why it was given this really unusual name, but what we identified was the fact that this protein is a negative regulator of wound healing. And we know that it is upregulated and is present in those wounds that Xanthe talked about, those chronic diabetic foot ulcers, those children with EB, those burn injuries, they all have really high levels of this protein. And what we know about this protein is that it increases inflammation, which leads to this chronic inflammatory response. And so the approach that we've taken is to try to reduce its activity, because we know that if we can lower the amount of phytolus present in a wound, then we can improve the way those wounds heal. So the way that we did with this was to develop some antibodies. So an antibody is just another type of protein that can recognize really specifically a protein of interest. And what it does is it actually binds to that protein and it neutralizes its activity, so it takes it out of the picture. And we have developed various neutralizing antibodies, and we've tested these in lots of different wound models. So if we inject these antibodies into the skin and into the wounds of mice and pigs, and we use pigs because pig skin is actually much more similar to human skin compared to the mouse, because mice, as you know, they've got lots of hair, they've got loose skin, and a pig is a much more better representative model of what a human repair process is like. So if we inject these antibodies into these different types of wounds, including burns and diabetic injuries, then we're able to significantly improve the way those wounds heal. As Xanthe mentioned, we're really focused on this devastating disease called epidermolysis bullosa, EB. And what we've been able to do is to develop a different way of treating that disease. So as Xanthe mentioned, not only do these children get wounds all over their bodies, but they also get this internal blistering of their intestinal tract. So what we did was to actually deliver the antibody into the bodies of our models, into our mice models. And we were able to show that antibody, when treated inside, was able to go to all the different wounds on the surface of the skin of the mice. We gave them multiple wounds. And we saw that there was a significant improvement in the rate of healing. But importantly, we also gave them IBD. So we fed them a chemical that actually causes blistering of the intestinal tract. And when we treated them with our antibody, we were able to really reduce, and that's the green bars here, compared to the control. And you can see that the severity of the IBD was significantly reduced. So we have the potential to be able to treat these children that will improve their healing of their external wounds and their internal wounds at the same time. Another project that Xanthe's actually been working on is looking at other ways to deliver the antibodies to burn injuries. So she's been able to incorporate these antibodies into a hydrogel. And that's just a gel that is very liquid, but it sets on the surface of the skin. And she's been able to show that if she applies the gel to the surface of burns on mice, that she can, and this is in 10 days, she can significantly improve the burn injury with those antibodies delivered in that gel. And finally, with the antibody project, we're working with a group at Monash and interested in diabetic foot injury. And what they've been able to do is to incorporate our antibodies into these porous silicon nanoparticles. And these are tiny, tiny particles that are full of holes. And we can actually get the antibodies inside the holes, and that protects them from the proteases, which are enzymes present in a wound that can degrade proteins. And what they've been able to show with us is that if you deliver these protected antibodies into a harsh environment like a diabetic foot injury, then we can improve healing responses. So that's all very exciting, and we've got lots and lots of really interesting data, but it's a long process between getting that laboratory research actually into a patient. And that's currently where we're working at the moment. We're working with various investors, and we're pitching our research to various pharmaceutical companies because we need them to start to get involved. So at the moment, we're humanizing our antibodies so we can put this antibody into humans, and then we're going to do some safety and toxicity studies, and then we're going to be able to generate the antibody in large enough quantities that we can do human clinical trials. So that's where we're up to with that. Another area that we're interested in is a cell-based wound therapy dressing. So many cells have been used for treating wounds, such as stem cells, and stem cells are cells that can actually change into different types of cells, which are important in stimulating that response. And we've been interested in trying to find ways to deliver those cells into people with diabetic foot injuries. So working as part of the Cell Therapy Manufacturing CRC, which brought together lots of researchers from industry as well as clinicians as well as academics, we've been developing a cell therapy, a surface that can be put onto a dressing that you can then apply the cells to, that can then be placed onto the patient, and the cells then go off and into the wound and stimulate healing. So we're using a process called plasma polymerization, and this is a fourth state of matter, so it's like your plasma TVs. And using this process, you can actually deposit chemicals onto a surface that enable a cell to be able to stick to that surface. But you've got to be really careful about the way that the chemistries are put onto that surface, because you want the cells to actually come off when they're in the wound. So we did a lot of work with a lot of material scientists to be able to really tweak this process so that we could finely tune the modification of the wound dressing, then apply the cells, and then test them using our in vitro assays and put them onto mice, and then eventually we put them onto pigs. And really interestingly, what we saw there was that this graph here on the left shows, in the grey bar shows how a normal pig wound would heal. When we treated it with our dressing, which had the cells on it, we were able to improve the rate of healing, so those wounds healed a whole 7 days before they would if they weren't treated. We were also able to show that we got 85% closure, much compared to the control. So this was all such important and exciting work that we've now done a clinical trial. So working with Professor Rob Fittridge at the Royal Adelaide Hospital, he's been able to put these dressings that we've developed with some industry partners, Cynata and TechSide, and he has run a clinical trial with patients with diabetic foot ulcers and shown an 80% improvement in healing, which is really significant and something that we're really excited about. Cynata is so excited by the data that they've now licensed the product from TechSide, and they're now moving that forward into broader scale trials around the country, and it'll be going around the world. So that's one really good example of how laboratory research can actually go all the way into clinical trials. Another advanced dressing that we've been working on with a group from our MIT is about nanodiamond silk dressings. And this is a really fantastic collaboration with biomaterial scientists, biophotonics, which is the use of light, with the wound healing biologists like Xanthe and myself, and then some clinicians. So it includes Fiona Wood, who's a brilliant burn surgeon in Western Australia. And what we've done there is to use these and develop these silk-based dressings that you can actually embed tiny diamonds into, which actually can measure temperature and pH. And by being able to, and those are early indicators of infection. Because as you know, if you get a wound and it goes warm, it means it's inflamed. So you can measure those tiny changes in temperature. And if you measure them early enough and sensitively enough, you can actually get in early with your treatment of antibiotics. So we're currently doing studies to show that we can monitor this change in temperature and really tiny changes in temperature, and also developing a handheld device that can measure the blood flow and the microvasculature as it comes back into the wound. So Xanthe earlier mentioned that we're really interested in diagnostics and being able to identify patients who might be at risk of wounds. So to do that, you need to think about what does a point-of-care diagnostic need to do. A diagnostic needs to be simple to use. It needs to be relatively cheap and not need any really specialized equipment. And you've got to have a really good biomarker that you can rely on that will change in a patient. And that's the key success for a point-of-care device. So as part of our project and the work we've been doing on flightless for so many years now, we identified that flightless is actually a really good wound biomarker. We did with a group in Queensland, we were able to use some data from a clinical trial that they had undertaken, where patients with these were diabetic patients, and there were several groups. They either had diabetes without a wound, or they had diabetes and didn't get a wound within 24 to 48 months, or they had diabetes but then got a wound within 24 to 48 months. So we're looking at, what happens to those patients before they even get their wound. And when we looked at the plasma, so we collected the bloods from those patients, and when we looked at those patients, we saw that even before they got their wound, they were starting to get really high levels of this protein in the blood. And that's most likely due to the fact that with diabetes, you get leaky blood vessels, and you get the release of these intracellular proteins that can then be measured in the blood. And we saw that we were getting an 800% increase in plasma flightless levels 24 to 48 months before that wound actually occurred. So this is potentially a really good way to be able to identify people who have got diabetes but don't have a wound, but may well get a wound in the future. So whenever they measure their blood glucose levels, they may also be able to measure their wound risk. So we're currently developing a diagnostic approach, similar potentially to what you all used to know as a VAT test. So this is just it's a process that separates proteins according to their size, and then we can come in with our antibody and measure the level of the flightless, and you'll just get a readout on your test. So a red line might mean that you're at risk of a wound, and then you will be able to take that to your GP or podiatry clinic or wherever it may be, and then we can start to do preventative measures to make sure that that doesn't happen in the future. The final thing I wanted to briefly mention are some wound technologies that we're developing, and only one technology is a photoantibody therapy that we've been developing now with a PhD student in my group, Nikki Black. And what Nikki's been doing is to look at whether or not you can use specific photosensitizers that link to an antibody that can then bind to a bacteria if it's present in the wound. So there's a lot of these photodynamic therapies on the market, and it's often used to treat infection, but it also can damage healthy tissue. So by being able to more specifically focus the light, which is what photodynamic therapy is, actually on the cells that you want to kill, so the bacteria, and not damage the healthy tissue around it, then you're going to get a better result. So that's something that we're currently doing. Nikki's just finishing her PhD at the moment, so we're looking to see what happens next with that project. And then I just wanted to, my last slide, just to say, it's not just UniSA who are doing this amazing work. There's lots of innovations around the world. And a lot of them are coming out of Australia, which we're really proud of. So we know that the University of Adelaide has done this amazing work on this temporizing matrix, BTM, which Xanthe mentioned. There's a company called Polynovo who are now marketing that to the world. It's a matrix that can be used in a skin graft in a burns victim, and it's able to help to reduce scarring of burns. And that came out of the University of Adelaide. We know that in UWA in Western Australia, Fiona Wood has been doing this marvellous work on spray-on skin, and she's been able to develop ways to culture cells taken from burns patients that can then be sprayed back onto the patient once the burn wound is starting to heal. And that's now being taken up by surgeons all over the world. In the University of New South Wales, they're developing antimicrobial glues. These are glues, synthetic glues that they've developed that can actually reduce bacterial from actually taking hold and forming wound infection. From the University of Wollongong, we've got 3D printing of skin. So they're actually able to manufacture skin that can then be put onto a patient. There's still quite a long way to go with that research, but it just shows the capability and what is potential. At UniSA, we've also got researchers who are experts in lasers and photo. I've lost the word, but what they're doing is to develop an optical smart bed sensor that can actually help to produce pressure injuries. So what they're doing is incorporating these sensors into mattresses, and patients who have to lie down for a long time, they can actually give an alarm if that patient needs to move, so it'll be rolled over, or if they fall out of bed. So there's really interesting developments going on there at UniSA. And then finally, CSIRO is developing digital telehealth, which is really important for our rural communities and being able to actually get more people aware of the best way to deal with wounds. So there's lots of innovations happening. So just to acknowledge that my group, Xanthe is a leading member of my group and the rest of the team, and we collaborate all over the country with people who are interested in wounds and biomaterials and all sorts of different people, which we couldn't do without. And obviously, we're funded by lots of different organisations. So NHMRC is a really good funder of our research. The Hospital Children's Research Foundation, Diabetes Australia. We also get funding from the Department of Defence in the US for our diabetic wound research. So we're very grateful for that. So with that, I'm happy to take questions, and Xanthe will come back up and perhaps take questions if you've got any. Thank you.
Thank you so much to both of you. We now come to the Q&A section of this afternoon. If you have a question for either of our presenters, perhaps you could just lift your hand and one of our team with the roving mic will get to you. Okay, we've got a couple up the back there.
Good afternoon. With the advent and interest in AI at the moment, I was just wondering whether you were working on the predictive capabilities of AI to combat the bacteria before they fester in the wound, just in inverted brackets, fester.
Do you want to take that one?
Yeah, so at the moment, AI has been incorporated in a number of handheld devices, not ones that we're working on. We were recently in the States at a wound healing society meeting, which involved clinicians and also large corporations, and that is certainly one of the things that we're wanting to do. So take it away from the individual surgeon and clinician being able to identify things and start to have some parameters around that. What we need to know is we need to know what to look at first. So one of the things that Allison mentioned is looking at those sensors that can detect really subtle changes. Once you've got really good data that you know that you're measuring, you can feed that into AI and then it gets automated. And that's a really exciting opportunity for the future.
Yeah, thank you. The protein flightless, why does the body produce it? I assume that in the right quantities it has some beneficial use.
Absolutely. So flightless is a really important protein that is present in pretty much all cells, and it's involved in regulating the cytoskeleton, which is the internal skeleton that every single cell possesses and is important in being able to make those cells move, proliferate, migrate, adhere. And it's also really important for initially kickstarting inflammation. So yes, it's definitely needed. But what we've found is that in instances where inflammation continues and is exacerbated, the level of flightless continues to rise and it isn't negatively regulated. So when it's present in too high levels, then we get continued inflammatory response, we get continued overexpression of various proteins that stimulate that scarring process. So yes, in small processes, absolutely fine. And we don't want to get rid of all of it. And that's also a real benefit of the approach that we're taking, which is an antibody approach, because we're not wanting to get rid of the flightless that is inside the cell. But what we're doing is mopping up all the flightless that has been released in wounds and is present outside of the cell. And that's what the antibody is then targeting. So we saw the way that we've approached it, which is serendipitous, because we didn't originally think that was the way it was going to work, but we're very happy that that was the way that it did work. We found that being able to neutralise the extracellular flightless is actually really important, so we're not getting rid of all that really important stuff that it does do.
Hello, thank you for your presentation.
Thank you.
My wife is currently in the RAH with chronic leg ulcers on both legs, and she's had them for about 4 years. She's dealing with the vascular department over there, Erica Crothers. Are you liaising with them about your latest research information for them to apply for the public?
So we work with a lot of vascular surgeons, but primarily with Rob Fittridge, who is the head of vascular surgery in the Royal Adelaide Hospital. So we work regularly with Rob, and he is aware of what we're doing. I guess we're quite early stage with the flightless project particularly, so we're still looking to get those into human trials. The cell therapy approach is much more advanced, that dressing that I was talking about. So Rob's already applied that to some patients, but we're now waiting for the company, Xanthe, to continue its product development so it can actually get it into the hands of the surgeons again. So as Xanthe mentioned, there's so many patients out there who need the kind of work that we're doing, and I wish that we could get it into them tomorrow. But there's a process that needs to be gone through first. But probably more raising awareness of those surgeons to let them know what we're doing would be great.
Thanks. What was that Xanthe product again?
It's called SciPatch.
SciPatch. Thank you.
Thank you. I was just wondering, the treatment you're looking at to reduce the internal in the intestine, those blisters and things, are you looking at perhaps sometime in the future, would it be effective for things like ulcerative colitis or Crohn's disease?
Yeah, should I go? Or you want to go? But absolutely we are, because, clearly there's a huge potential for that in treating patients with ulcerative colitis or IBD. And we've done studies that show, in patients with IBD and shown that flightless is present at high levels in their guts. So absolutely, the work that we're doing could have real importance for treating those patients, as well as these, children of EB. So, yes.
Thank you for your talk today. This might show my ignorance, but people who have passed and have donated their body to science and medicine, has there been a technique developed, or is there any information about skin grafting from a deceased person to a living person who is in need of skin grafting?
Primarily what's used if you do donate tissue to science can be used for our in vitro study, so in the lab where we're looking at investigations. What you want for grafting is you want really viable cells, right? So potentially you could do that very quickly. However, quite often what happens in the donation process is it would then get decellularized. So we'd use it as the structure to then test and put live new cells in, and that's the approach. And I've done some work in our lab where we do use 3D skin in the lab, and that is based on that. There's issues when you transplant from what's called not from your own body. So certainly there's a big push towards having, taking patients' own cells and getting them to grow up so that then you can return them, because you get less rejection issues. So as far as I'm aware, it's not really an approach that is taken by having a look at grafting from cadaver grafts across, but they're certainly still very useful in the process of investigating. So one of the things that you've seen here is that there's so much that we have to do in the lab before we get into the preclinical models, before we can get into the human trials, before we can get it to market. So it's definitely useful.
I've got another question. I'm just wondering how is Australia situated in the event of another pandemic, or, for want of a thing, they keep on talking about world wars. Are we able to produce these dressings and medicines ourselves, or do we have to import them?
That's a really good question, because we currently don't have a great sovereign capability in a lot of this manufacture, and there's a lot of interest and work being put into seeing if we can develop better industry, better ways of getting industry to work with universities and being able to develop that. It's the biomanufacturing process that is often has to be done overseas, and then you do have a problem bringing it back to Australia. So it's still an emerging industry, it's still an emerging sector that needs a lot more funding and a lot more support. So, yeah.
Well, that just about brings us to the end of today's seminar. I'd like to thank both Allison and Xanthe for coming to speak with us today. Thank you for such a fascinating presentation. We do have a small gift for you as a token of appreciation for your presentation today, and Amanda's got that for you.
Thank you very much.
Thank you.